So I am Dr. Christopher Pena. I am the scientific liaison at the University of Chicago Comprehensive Cancer Center, and I am here with Dr. Mekinda.
Hi, I am the Assistant Director for Cancer Education, also with the University of Chicago Comprehensive Cancer Center. And Dr. Pena and I are here today with students from our Chicago Eyes on Cancer and Research Start programs. And these are summer cancer research programs for students who think that they might be interested in a career in biomedicine. So our students work full-time in the laboratories over the summer with university faculty. And they are developing research and career skills, but importantly, they're contributing to this cutting-edge research in cancer and cancer care. It's really helping us to understand cancer and how to end it.
So these students are incredibly excited to share their work with you today, and this is just part one of a four-part series. Today's episode will focus on cancer treatment. The second episode, which is a two-parter to this one, will focus specifically on cancer immunotherapy. The third episode will focus on cancer detection and prevention. And the last episode will focus on survivorship, which is basically living after you have been diagnosed with cancer and receiving cancer treatment and the certain types of lifestyle changes that are associated with that.
So we really hope you enjoy these programs, and we really hope that you are very impressed with our students, because they are in high school and college, but already they are skilled researchers and they are talented scientists, and we truly believe that they are the next generation of cancer researchers and care providers, and we cannot wait to see what they do.
Excellent.
So with that, I will say goodbye and make some room for your guest.
Thank you. So again, we are Chicago Eyes on Cancer and Research Start. We are a program for high school and undergraduate students, and we give them the opportunity to do summer research in a cancer biology lab. I am here with my first guest today. If you want to welcome Crystal. How are you doing?
I'm doing good. How are you?
Good, great, thank you. So why don't you start off by telling us exactly what is cancer?
All right, so cancer occurs when cells in the body become abnormal. So they divide out of control and they acquire the ability to spread into surrounding tissue. And so people more commonly know about cancers when they refer to tumors. So tumors are an abnormal mass, abnormal mass of cells. And they could be benign, which means they're not cancerous, or they could be malignant, which means that they are cancerous.
So I'm always hearing that there's a new breakthrough here and there's a new breakthrough there, but ultimately it seems like we still haven't cured cancer yet. So why is that?
So it's important to note that cancer isn't just one disease. There's hundreds of types of cancers, and even the very big ones, such as prostate cancer or breast cancer, etc., they have different subtypes within them. So each of these cancers are so unique in their own way that they have their own methods of treatment, which are distinct from one another.
So what exactly do these cancer treatments look like?
So, as I said before, because cancers are so complex, for example, they're dramatically different in the way that they show up. They're also different in the stage of cancer that they are, how advanced they are, and also they have different genetic changes within them. Not all cancers are the same. And so some cancers, for example, can be treated with radiation therapy. Meanwhile, others might need chemotherapy. Or in some cases, cancers could be surgically removed. And there's also more specific targeted methods of treatment that have been developed as science has progressed. And so, for example, there's personalized methods of treatment which target the specific aspects that are unique to a person's cancer. For example, in my lab, we work with analyzing tissue composition levels in prostate cancer, which we use in order to see what's the best method of treatment for our patients. So we look at the Gleason score, which in prostate cancer, it's basically like the grade of cancer that it's at. And through that, we're able to see, okay, well, how far along is this cancer? Will this cancer cause the patient to die? Can they live with it? So that they don't have to go through things such as over-treatment, which is a very big problem in the treatment community.
Great. Well, thank you so much for sharing your research with us. I think we're going to get our next guest here.
All right.
So again, we are Chicago Eyes on Cancer and Research Start. We are a program for high school and undergraduate students that want to take part in summer research in cancer biology. You will see the link on the screen right there if you're interested or know somebody who is interested that wants to sign up, or you can call us at 773-702-4678. I am here with my next guest, Linnea, and you are going to tell us exactly how we are trying to improve cancer treatment.
Yeah,
Yeah, so I specifically work in skin cancer. There's a lot of different types of cancer, but I work in skin cancer. Skin cancer is a very common cancer in the United States as well as all over the world. The number one cause of skin cancer is, of course, UV radiation, and so UV stands for ultraviolet light. This radiation you're exposed to via sun rays or tanning beds, situations like that, right? Mm-hmm. And so my specific work focuses on understanding how a fat mass and obesity-associated protein called FTO is involved in a cell's response to UV radiation. And we're also interested in looking at how the cell's apoptotic pathway happens after UV radiation exposure, because when investigating skin cancer, it's really important that you understand how your cells, specifically your skin cells, are affected by this UV radiation.
So does this mean you work with patients since you are, in fact, working with treatment?
So no, I don't actually work with treatment, or with patients, sorry. In cancer treatment, there's a lot of different jobs that people can do. So some people work in the clinic, and I think that's the most common way that people assume cancer treatment happens. But there's a lot of people who work in wet lab settings, which is what I do. And so I work at a lab bench or in a tissue culture hood, running experiments, doing tests, analyzing data, things like that. And so specifically, I work to run tests to decide whether or not FTO is involved in this UV radiation process or response. And then I also run tests to understand what kind of proteins are involved in this apoptotic pathway after it is begun.
So if you're not technically seeing patients, how does what you do help them in the long run?
Yeah, that's a good question. Understanding the pathways that your cells undergo after these cancer-causing stimuli is incredibly important in the field of cancer treatment, right? Whether you're determining what's causing these pathways to undergo in the first place or what's part of the pathway once they do begin, really not only helps you understand the cancer better, but also how to treat the cancer better. So, understanding how FTO prevents or aids in the beginning of the apoptotic pathway is really important, as well as understanding how or which proteins are involved in that apoptotic pathway, because you want to be killing the cancer cells. So, understanding how these pathways work and what causes them is not only important when understanding cancer, but also when understanding how to treat it better.
Great. So in the field, we always hear something bench to bedside, and it sounds like you're working at the laboratory bench, looking at all these different pathways and ultimately how understanding them will be better for patients in the long run.
For sure.
Great. Well, thank you for stopping by.
Yeah, thanks for having me.
Thank you, Linnea. So, again, we are Chicago Eyes on Cancer and Research Start. We are a summer program that is geared for high school and undergraduate students, and we give them the opportunity to do research in a lab specifically pertaining to cancer. If you are interested in joining this program or you know somebody who is, feel free to check out our website, which is listed on the screen. You can also reach us at phone number 773-702-4678. I am here with my next guest, Rene. How are you doing?
Hi.
Good. And you're going to tell us a little bit about ultrasound today. So what exactly is ultrasound?
Yeah, so ultrasound is truly a beautiful thing. Some people think that there's radiation involved with it, but actually it's just mechanical sound waves bouncing back and forth from the machine and the human body, whether it's organs or tissues and things like that. And ultrasound is most commonly known for imaging purposes for pregnant women, for fetuses. And also, a lot of doctors like to use ultrasound during biopsies and surgical procedures and things like that. But ultrasound can actually also be used as a treatment therapy, as a targeted treatment therapy.
Great. So can you elaborate on that a little bit, how ultrasound can be used specifically for cancer patients?
Yeah. So the method that my lab is working on at UChicago during this internship is actually called histotripsy. And what that is I like to think of it as, you know how you can burn ants with a magnifying glass using the sun's rays? It's sort of like that. You target a very specific area in the body, and you just send these sound waves back and forth at very high pressures. And what it does is that it ablates tissue in the body. And ablation is just a fancy word for tearing up and destroying tissue in the body. And so when we actually focus these sound waves onto this specific area, it creates these bubbles inside the body. And when these bubbles collapse, these tiny little molecules called reactive oxygen species are created, or ROS for short. And these are really the tiny little soldiers that go on and destroy and ablate that tissue. And after that tissue is destroyed, they sort of just retire and die off.
Wow. So what's really interesting, it sounds like you're using sound waves to introduce these ROS species inside a patient. That does sound a little scary. Do you know if there are any trade-offs with this method in terms of being harmful to the patient? Are there any side effects that are associated with it?
Yeah, so histotripsy is currently in its preliminary studies, and what that means is that we're just fresh off the start for doing these trials and things like that. So as of yet, we haven't seen any major side effects or any factors that would decrease the quality of life of the patient. But really, the good thing about histotripsy is that this is ultrasound, this is not radiation.
Right.
And radiation truly, we all know that there's these side effects with it. And using ultrasound, we want to, it's non-invasive. So you're not cutting people open and you're not invading the body. And it's also safe because you are generating these things within the body and it stays within that area. So as of now, we don't see any major side effects, and we just hope to push this out into the market to better the lives of many patients.
Great. That sounds really awesome. The fact that you may not even need surgery at some point, and something that isn't as harmful as radiation. So keep up the good work, and thanks for doing some research and sharing it with us. We're going to go with our next guest, but before that, again, I want to stress we are Chicago Eyes on Cancer and Research Start. We are a program for high school and undergraduate students that want the opportunity to do research in the lab, specifically in the field of cancer biology and oncology. Our website is on the screen right now. If you know someone who's interested, or if you're interested in signing up, you can also reach us at phone number 773-702-4678, and we'd really like to see you in our program. So I'm here with my next guest. This is Zak. How are you doing, Zak?
Good, how are you?
Great, thank you. So why don't you share with us a little bit the kind of what kind of cancer you're studying specifically?
Yeah, so today, or this summer during my lab, I am working with glioblastoma, or Glioblastoma Multiforme, which is the most common type of malignant brain tumor amongst adults. So basically what that means is glioblastoma is one of the very aggressive tumor that is in the brain. And this means it's very hard to treat, and also results in very low survival rates in patients.
Wow. So why is glioblastoma really hard to treat?
Yeah, so there are many different reasons why glioblastoma is very hard to treat. One of them being the blood-brain barrier. So what the blood-brain barrier does is it carries blood to the brain and also to spinal cord tissue. But another thing that it does is it keeps any foreign objects or substances from entering the brain. So when you're trying to administer a drug to glioblastoma, or the tumor, it's very hard for the drug to get through this barrier because the body thinks of it as a foreign object and not as something that's trying to help it, so it automatically attacks it, which makes it very hard to treat glioblastoma.
Wow. So, I mean, with this blood-brain barrier kind of preventing a lot of these treatments, are there any kind of drugs that can cross the blood-brain barrier?
Yeah, there definitely are. One of them, one of the most common being temozolomide, or TMZ, which is used after surgery in conjunction with radiation. And basically what it does is it goes to the tumor and it suppresses the growth. But when it crosses the blood-brain barrier, its efficacy is kind of hindered, so it's not as effective as it would be.
Wow. So are you working with TMZ in the lab, or can you share a little bit more with us about your specific laboratory research
Yeah,
experience?
so during my research project is primarily focused on temozolomide and glioblastoma, not so much the blood-brain barrier, but how temozolomide is used in treatment. So there was a past experiment done with temozolomide where U87 cells, which are basically just glioblastoma cells, were treated with different concentrations of temozolomide, one being starting at 0, so no temozolomide, and then increasing in concentration from there. And what we saw was in the cells that had higher concentrations of temozolomide, there was this certain protein that was being expressed, CDK1, which is part of the CDK family, which are cyclin-dependent kinases, which promote cell growth and the cell cycle. So basically what they found was temozolomide was actually causing this expression of this protein, which actually led to tumor growth and mitotic progression. So what I'm doing is I'm basically replicating that experiment in GBM44 cells, which are patient-derived glioblastoma cells.
Wow.
So to actually find out if this is actually happening within a patient.
Wow.
Yeah. So after I find my results, some future directions could look at finding ways to suppress CDK1 expression in order to make temozolomide a
More
little
like effective.
Yes, and yeah.
Great. So that's really interesting in the sense that you have a therapy out there that initially works, but then it can make the cancer a little bit stronger, and so you're trying to figure out a way to stop that part from happening.
Yeah.
So thanks for sharing that with us.
you.
Really great work you're doing. Thank you. So again, we are Chicago Eyes on Cancer and Research Start. We're a program for high school and undergraduate students that want the opportunity to do laboratory research in the field of cancer biology. If you are interested in joining this program or know somebody that is, we have the website on the screen for you right there. You can also reach us at phone number 773-702-4678. I am here with Linnea and Crystal, and you guys are going to tell us a little bit more about what your experience has been like in the lab. So why don't you go ahead and share with us that?
You want to get started, Liz?
Yeah, my experience in the lab has been great. I think most people would agree that their experience has been great. I've really loved my research specifically in cancer. I never really thought that I would be as interested in cancer research as I have become because of this program. I always had a baseline interest in it, but I mean, I've only been in the program for a little over a month, and it's been insane. It's been great. I'm really excited about cancer research, more than I ever thought I would be, and I definitely will continue in the field from here on out.
Great.
Yeah, so for me, this is going to be my second year in this program, in the Chicago Eyes on Cancer program. Formerly, it was known as the CURE program when I applied. And so for me, I've been in 2 different lab settings. So last year, I worked conducting more qualitative data analysis. So I would call patients and I would check up on them and collect follow-up data, which was completely different than from what I'm doing right now, because now I'm working more in a setting where I'm measuring tissue composition levels, which is more computational work that I'm doing with a specific software that we use. And so for me, it's been interesting to work in those 2 different lab settings because I get to see, what I do like and what I don't like. So what I've definitely noticed is that I enjoy working in a dry lab, which is not in a wet lab, such as what you work on, like sitting on a bench. I feel like it takes a certain type of person to work in a
So what
wet
is
lab.
the difference between a wet lab and a dry lab?
So I think you could explain what a wet lab is.
Yeah, so in a wet lab, basically that means that I'm working with cells, I'm working with chemicals, I'm growing the cells, I'm extracting the cells, I'm doing tests on the cells, and that means a lot of different things, right? So that means that I'm like physically working with the cells, but after I've collected them, it just means like running them through a machine and then interpreting data, right? But yeah, I am physically, in the lab, gloves on, working with the cells, working with the, tissues and stuff like that.
Yeah, so I guess a dry lab is basically, for me at least, it's been sitting in a desk working on a computer, and for me, I might be receiving data that's from a lab and I might be analyzing it, and so that's more what I do in comparison to a wet lab. So personally, I've never been inside a wet lab, but they're very cool.
Cool. So do you actually get to work with either like patient cells or patient data from like real cancer patients?
Yes.
Wow.
So for that, we have to have certain specific type of training. For example, when we first when we first get into the program, we have a week of orientation. And during that week, what we do is we conduct HIPAA training, or for example, later on, once you actually go into your lab, they may ask you to conduct further training. Like for me, they're asking me to do like CT training, which is just further like patient confidentiality training that you have to do.
Right.
Yeah, so my lab works with mice models, and we also work with human cell lines. So.
Wow, that
Yep.
sounds really interesting. So, what has been the most rewarding thing that you've taken from this program?
I guess two things for me. I think in any new setting, especially when you're working in like cancer research, you're gaining a lot of skills, right? So even if you've worked in a lab before, you're going to be doing different tests or working with different samples, and so those require a different set of skills, different techniques, different protocols. And so by being in a new lab setting, you learn to do more things, you learn to do more tests, you learn to do different experiments, which has been really great because, as a researcher, someone who wants to pursue a career in science, like the more exposure, the more techniques you learn, the better you're going to be and the more well-rounded you're going to be. And so that's been really great. It's also been, I guess, the people in the program, not only the directors or, the people who are working both behind and front of the scenes to make this program happen, but it's also been the fellow participants. I know we all work in different labs, but we go to lectures together. We do fun outings together too, and that's just been really great because they're all really great people, as you can probably tell. And it's been really fun, and I've made a lot of friends, so that's really nice.
I think for me, one of the most rewarding things was definitely being able to say that I'm in a program like this, because for me, I'm still in high school. So when I got into the program, I was 16. And so for me, I was very overwhelmed when I heard that I was got in, because I was emotionally crying tears of joy, because the University of Chicago was one of my dream schools, and it was a really big honor for me to say that, at 16, I was able to work in a lab and I was able to conduct cancer research. And for me, that was a really rewarding experience. And also the fact that I could go back and people would come up to me and talk to me. So, for example, I had a girl that she came up to me and she told me, oh, well, my mom had cancer, and I really, admire what you're doing. And to me, that really made me happy to know that although I didn't work in the specific cancer that her mom had, she really appreciated what I was doing, and she appreciated that someone was interested in what was happening in her mom's life, and also how it affected her.
Great. And so we have a little bit of time left. Do you want to share any last minute thoughts about the program or your experience in general before we wrap up?
Start this one.
All right, so just last minute thoughts. I encourage anyone who's watching to apply or to look into. It's not only open for undergraduate students, but it's also open for high school students as myself. And so I really encourage anyone to apply if they want to gain more experience. Also, you don't have to necessarily have to be interested in research. For example, for me, some people might go into this program wanting to be a PhD like Linnea, but for me in high school, being in high school, I was like, oh, I want to be an MD. And I think that's what most people think starting off in their career when they want to go into the medical field. I think maybe I'll be an MD. But being in this program has taught me that there's various different paths to take in the medical field. For example, you could be a PhD and you could be doing research that is contributing to benefit in other people's lives. Or you could be an MD, which is working more with people. Or you could be an MD PhD. There's so many various different things that before I didn't know. And so for me, it really opened my eyes to the other career opportunities that I have.
Great. Linnea?
Yeah, I just kind of want to echo what Crystal said. I think that's a really good point. But there's also people in this program that are computer science majors or interested in optimizing the tools we use in lab or the machines. And so you don't even really have to be interested in getting your MD or your PhD, but just interested in the field of cancer itself and all that entails, because it is such a wide-ranging field that I think affects probably everyone in the world. I mean, we all know someone who's affected by cancer. And so I think there's a lot of opportunities through this program. It's a great program, super fun. You learn so much. And I think anyone can get excited about this great program.
Well, thanks for sharing all that with us. Just to wrap up here, again, we are Chicago Eyes on Cancer and Research Start. If you follow the link on the screen right there, if you're interested in applying or know somebody who wants to apply, you can also reach us at 773-702-4678. And you'd hear great stories and great experiences. that we have with our students. So just wrapping up again, and we want to thank you guys for tuning in and thank you guys for sharing your experiences with us.
Thanks for having us.
Alrighty, thank you.